Hello, let's continue talking about organic molecules, and now we're going to talk about proteins and nucleic acids. So proteins are by far the most abundant and important organic molecules for your body. In fact, if you remember, last time we mentioned briefly that DNA codes for proteins, which means all of those genes that you have in your chromosomes in your DNA are only telling your cells how to build proteins. So even though you make things like glycogen for storing carbohydrates in liver and muscles, there aren't any genes that specifically tell you how to make a glycogen molecule. Instead, you have genes that tell you how to make a protein enzyme that can then build glycogen. So proteins are extremely important, and there are many, many different types of proteins and therefore many different functions in the body. Now, the basic building blocks of proteins are amino acids. Now, this is important because you cannot make amino acids. You can make proteins. We said before that your DNA tells you how to do that, but you can't make the ingredients for proteins. You can't make these. You can, however, make glucose. And we haven't really talked about that yet, but that will come up over and over again in the semester. Your body has the ability to make glucose molecules from non-carbohydrates if you're not consuming enough carbs. So if your body, for example, goes through some period of prolonged fasting, your body will use up stores that it has, and then it will go into a process called gluconeogenesis, which is a fancy way of saying creating new glucose molecules from non-carbohydrates. But you can't make amino acids. So you have to make sure that you have some source of protein in your diet to make sure that you're getting those building blocks. Now, there are a lot of different types of proteins and a wide variety of functions. We're not by any means going to be able to cover all of them. So what I've done is broken it down into 7 kind of categories of functions for these proteins. We'll talk about each of those and give some examples of specific proteins that fall under those functions. So we have proteins that provide support. So these would be things like the proteins that you find in hair or fingernails, called keratin. We have proteins that provide movement. In your muscles, you have proteins that reach up and grab another protein and pull it across so that your muscles kind of slide across each other to contract. So those would be things like actin and myosin. We have lots of different types of proteins that help us with transport. Albumin in the blood is a protein that binds to other substances to help transport it. And one that you're probably a little bit familiar with would be hemoglobin. Hemoglobin is found in your red blood cells. It binds to iron, and then that iron binds to oxygen so that you can make ATP. So this is a very important protein. We have proteins that help us with buffering. We talked about that before, and that's to help us maintain our normal pH of our fluids. And if you remember, the blood pH range is 7.35 to 7.45. We have proteins that help us with metabolic regulation. These would be things like enzymes. A good example of that would be lactase. Notice it ends in ASE. Not all enzymes end in ASE, but many do, and they often tell you what they're acting on. So lactase helps us to break down lactose. We'll talk more about those later on. We have proteins that help us with coordination and control. So we have protein-based hormones. A good example of that would be insulin. And then we have lots of defensive proteins, in particular antibodies. Now, antibodies are in the news a lot, and they don't really attack a foreign substance and destroy it. What they do is they grab a hold of it and bind it and basically flag it for destruction. So you can think of antibodies like those little kind of like yellow flags or orange flags you might see tied around the trunk of a tree to let the tree crew know that those are ones that they need to cut down. And that's basically what antibodies do. So they mark a foreign substance or an infected cell as something that's foreign, and then your white blood cells can come and destroy that. Another good way of thinking of antibodies is to think about the Incredibles movie. So if you think about the first Incredibles movie, if you remember, Mr. Incredible broke into Syndrome's lair. Now technically he was breaking into Syndrome's house, right? So think of that house as a cell, and Mr. Incredible was the bacteria or the virus that broke into it. Now as he was trying to escape, the guns came out and shot him with big kind of black balls of goo. And those balls of goo stuck to him and held him down until Syndrome, the white blood cell, could come and take care of him. So those black balls of goo were basically representing antibodies. Now, proteins have very complex structure to them. They all have kind of this basic structure down here that's the same. And then they have these other little, what we call R groups, or functional groups attached to them. And these R groups, or functional groups, give that protein or amino acid different characteristics. So when I link these different amino acids together, I have these different R groups that are going to potentially interact with each other. And when I link the amino acids together, they're held together by a special kind of covalent bond called a peptide bond. Now in your stomach, you have enzymes that break down proteins, and that enzyme is called pepsin because pepsin attacks peptide bonds. Now proteins have very complex structure. The primary structure is the sequence of amino acids. So it would be this amino acid attached to this one, attached to this one, and so on. And then these are those peptide bonds. The secondary structure is that we end up getting kind of these pleats, like a pleated skirt or a pleated curtain, or we get these spirals, these helices. And then we get the tertiary structure, which gives us our final kind of three-dimensional shape of the protein, where these different R groups interact with each other. So let's say we have a couple R groups here. Imagine that this one is positively charged and this one is negatively charged. That's going to make them attracted to each other. So we end up getting all this really complex folding of our protein to give us our final shape. Now that might be the end of building our protein, or we might have quaternary, or a fourth level of structure, where we have two or more chains connected together to make a final protein. Hemoglobin is a good example of that. It is composed of 4 different proteins that are all linked together. Now, if I do something to change the shape of my protein, I could end up with something like this, like sickle cell. Hemoglobin, if there's a single base pair mutation in the DNA that codes for hemoglobin, we can end up with getting a protein that ends up kind of being shaped all funny, and we end up with these odd-shaped cells. And they're more likely to get stuck to each other and form blockages or clots. We'll We'll talk more about that when we get to blood. Now, if I change the shape of that protein, I can denature that protein. So you can take my functional protein, I can heat it or treat it with a different kind of pH, like an acid, and then I could end up permanently changing the shape of that protein. So if we take something like albumin, which is found in egg whites, they're kind of clear and runny. But if I heat them up, I end up with this white kind of rubbery substance. So I've permanently denatured those proteins. You can sometimes, however, temporarily denature a protein. So I can take a protein like the proteins found in milk, and then I can denature them with heat, but then I can cool them back down and they can kind of reform. So they kind of become untangled and then they kind of reform. Now the last thing that we need to talk about with organic molecules are nucleic acids. Now we're going to talk more about nucleic acids when we get to cells and we get into DNA in detail. So for now we're going to keep it really simple. The examples for nucleic acids that you should be familiar with are DNA and RNA. Now their functions, if you remember I said that DNA is basically a giant cookbook of protein recipes. And each of those recipes are genes. So a gene is a recipe for a specific protein. Then the RNA molecules would basically build those proteins. So they're kind of like the chefs. And we'll get into the specific types of RNA in a lot more detail when we get to cells. You don't need to worry about the differences between the types of RNA for the chemistry test. So what's the relationship between nucleic acids and proteins? Nucleic acids basically tell you how to build these guys, and then they build them. So nucleic acids tell you how to build proteins and then actually put those proteins together. Kind of ran out of a little room here, didn't I? So if I look at a nucleic acid, we have a polymer or a long chain of nucleotides linked together. So these are the building blocks for nucleic acids. So let's kind of take a look at these. So first, if we look at DNA, we said that it's a big cookbook of protein recipes. You inherit these genes from your parents, which are the recipes. Where would DNA be found? Be found in the nucleus of the cell. And as we mentioned before, the coding regions that code for a specific protein are called genes. So you've probably heard of chromosomes before. So if I have one long chromosome, I'm going to have sections along it called genes. And each of those genes is going to code for a different protein. So as we mentioned, nucleic acids are basically storing and processing information, specifically information on how I make proteins. Now you don't have to be able to spell out the long name for these, but it's helpful for knowing the structure because it tells you right in the name that DNA is a nucleic acid and that so is RNA. So if we look at a nucleotide, it has three parts to it. You have a sugar, a phosphate group, and you have some kind of nitrogenous organic base. The bases are going to vary depending on whether you have DNA or RNA. So let's take a look. If I look at RNA, we have a single strand where we're linking sugars to the phosphate of the next nucleic acid, nucleotide, and so on. So this is a nucleotide, this is a nucleotide, and so on. If I want to build DNA, then I take a second strand and I have to link them together using hydrogen bonds that we talked about before, because those are really easy for us to kind of unzip and break apart so that we can copy our DNA when we're making new cells. Now, DNA has 4 different bases in it. We have A, T, C, and G, or adenine, thymine, cytosine, and guanine. And there's a rule for which ones pair together. You have A and T, and G and C. So if this is an A, this would have to be T. If this is G, this would have to be C, and so on. RNA is a little bit different. It's going to have adenine, and it's going to have cytosine and guanine. But instead of thymine, it has a nucleotide called uracil. That'll be important when we're talking about actually reading the DNA and building proteins, which we'll do when we get into cells. Now, there's one specific type of nucleotide that we've already been talking about. We just didn't say that it fell under nucleic acids, and that is ATP. So we talked about this as being our main energy-carrying molecule. So these are the Duracell batteries that are running all of those different functions in the body. And we have a base, a sugar, and a phosphate group. So that sounds like a nucleotide. But what makes these special is that we have 2 extra phosphate groups attached to them. And these bonds out here, in particular this one, are really easy to break, and they're very high energy because they're difficult to put together, so they want to come apart, and then when they come apart, we get lots of stuff out of that pixie stick. So ATP is a modified nucleotide. All right, we're going to end there for today, and there's a video for you to watch on enzyme function.